session on emerging areas of eye research. We have 7 presentations for this, and I think we are missing one of the speaker. Couldn't turn here up because of visa issues, and so we have 6 of them. We're waiting for Dr. Shobha. Meanwhile, I take this privilege to call Dr. Calvin Pang. Is very much known to Indian eye research diaspora, in which he got the highest honor of Indian eye research group in 2014-15. He got the Balasubramaniam oration. So he's well recognized and well known to all of us. So I'm happy to listen to his talk on light therapy of children with myopia. Calvin, please.
Thank you.
Okay.
So the second one. Thank you very much. Very, very glad to be here. And I'm one of the organizers, so I thank the organizer as well, right? But it's what our chair said. I'm very much acquainted or be friends of Indian colleagues, right? And before COVID, I used to come to India from Hong Kong almost once every year, right? I have visited and have exchanges with most, if not all, major centers in India. I can tell you, in India, I've been to Chennai, of course, a few times. I've been to Madurai, I've been Hyderabad, right? For of course, Elvi Prasad and Aravind, and Chennai's Sakura Levalea, been to Mumbai, have been to Bangalore. So quite, a wide, travel in India. So it's always a great pleasure to visit India again. So after the COVID, it's been 6-7 years, right? So back to India. So I'm very, very honored. Unfortunately, for this conference, because of visa issue, some of us know that. So some of our speakers cannot come, right? And we probably have to miss, right? So I'm very glad to come here to share with you some of our work in Hong Kong. Now, I particularly chose this topic for our audience here because light therapy is one of our latest work, right? So we haven't done this for too long yet, about a year's work only. But some of you may know that in children myopia, we've been working on this quite rigorously over the last 10 years, right? More than 10 years now. And we have been publishing a lot of data that we have collected on Chinese children in Hong Kong, on the phenotypes, and also we do quite a lot of genetic studies on myopia. But then this theme of today, this section is emerging areas of eye research. So I purposely share some of this work with you all, right? But for us, light therapy of children myopia in Hong Kong, we are still at the early stage. So I'm very glad to serve you what has been done before. So myopia is known to all of us, I'm sure, here. Now, on one hand, it should be very simple. Just talking about essentially the elongation of the eyeball. Essentially, of course, there may be some other problem with the cornea, with the muscle, but essentially is the focusing problem. So it could be very simple, right? You could have just, a transcription factor. You could have just a growth factor with some derangement, and you lead to this, unbalanced growth of the scleral. Right? However, it is not like that. So on one hand, it could be simple. On the other hand, it could be very complicated. That to lead to this elongation eyeball or some other, problems that lead to the focusing of subject in front of the macula instead of, right at the retina. So we know that it is very complicated. And that's why even today, we are really struggling in treating myopia. So we all know in this audience that we need to prevent myopia, particularly children. We all know it's very, very serious. Say back in Hong Kong, 95% of our university students are myopic, essentially exomyopic, essentially, right? And even our children, right? We're talking about 30% of our primary school, young primary school pupils are myopic. And we know that childhood myopia would just get developed. So we are very, very eager to find ways to prevent myopia, right? So if we get to know people in the field, essentially right now we have 5, actions that we can take. One is very well established now, increase outdoor time. Now, outdoor also means in daylight, right? So I think this is understood, right? Outdoor time in daylight is not activities outside. It's the time spent outside or outdoor daylight. So we have recommendation for that, right? At least say 2 hours per daily. So this is based on a lot of population in Hong Kong, China, Asia, India, America, Australia. So now the small prints that I give here are the conclusions so far of what's recommended to prevent myopia. Increased outdoor time is very established. And of course, good reading habit. Essentially, need not avoid near work and not too long near work, in a distance of 30 cm, right? So again, we have worked out some sort of recommendations, not too long, for less than 30 seconds, not too long. Means not too near, within 30 cm. That's what this distance we're recommending. And of course, we always advocate regular eye check for our children, right? So that's what we want. We want all parents to be aware of the eye health of their children and regular eye check. But then this is not simple, because regular eye check also involves a public health service, right? It involves optometrists, it involves ophthalmologists. It's not as simple as it seems in some other places. I think in India you're doing great that your children can have regular eye check, but that's very, very important. And there are optical methods. And we know about that, those so-called defocused spectacles or OK lens or full keratology lens. And some of our friends know that, say, in Hong Kong, we advocate the use of 0.05%, 0.05% atropine as a low concentration atropine. Now, atropine have been used to retard the growth of the eyeball for some years now. And then there are different concentrations. And based on our data in children in Hong Kong, we advocate this 0.05%. There's a balance between the efficacy and safety. So these are fairly well established, right? Although we still need a lot more data, more multi-center studies to confirm all these, methods of myopia prevention. But we still need alternatives, right? So recently, the light exposure therapy myopia control have been under good study. And that's why I said, even in Hong Kong, in our department, we also have started this about a year ago. But there have been some very important work that have been published. And also light therapy and combination therapy. Now come back to increased outdoor time. It's very established. This is a phenomenal paper by Ian Morgan and Kathy Rose, right, published based on their work in Australia in 2008. That's well established that they, based on their data, and of course later validated by many other studies, which I refer to very briefly, is high level of total time spent outdoors that we should add in daylight, right? Other than especially engaged in physical activity. So it's not outdoor activity, it's outdoor time in daylight. That's well established now based on this pioneering study, and the fact is there already. And in China, I have selected 3 randomized trials, very well established. The first one, of course, there was very high profile publication by Minggong He in JAMA back in 10 years ago now, right? There is a randomized clinical trial on the daylight and primary schools in Guangzhou, one of the major cities in the south of China, right? So that's very clear to establish outdoor activities. And then also, of course, in Taiwan, particularly by the work by Professor Pei Chang Wu, And in Kaohsiung, in particular, randomized intervention clinical trial, again, on grade 1 young children aged 6 years. Very, very nice control study. And of course, later, also in Shanghai, again, Shanghai is one of, if not the biggest city in China, prospective, this time is prospective, cluster randomized examiner mass 3-arm trial involving more than 6,000 children, 6 to 9 years old. All in all, the conclusion is, we all came down to similar conclusion, a different design study, that extra 40 minutes of outdoor time reduces the incident myopia by up to 23% after 3 years old. Based on that, it's very, very clear now. But then controlling myopia progression is perhaps less related just on outdoor time. So because of that light, right? Outdoor time is outdoor light. They come in, so all development go to sort of focus on laser, on the special light, right? On the sort of light exposure. And then based on animal work, that is we found that it's repeated low-level red light. So I'll cut the long story short. It is low-level laser, wavelength 650. That is the optimum for dealing with myopia. And this is just one example of the system that been using now the prototype, right? And there are different designs. Essentially, is that you have a child very comfortably put the eyes onto this machine, and 20 minutes or 30 minutes in a day, and then they have standard protocol, irradiation 650 nanometer, right? I would say this is, if not the first, then is one of the very important pioneer work, again by Mingguang He, on the repeated low-level red light therapy, right, for myopia control. This one conducted also in Guangzhou, 246 children, very, very well designed study. I can speak longer and all that because we have less speakers now, right? So I can be a bit more relaxed, and we can hopefully get to know a bit more about this topic, and so are our other speakers. So these are the results, and leading to axial length shortening at 12 months with this repeated low-level red light, definitely reduction. So to be quicker, this is the conclusion by this very important work, that when these Chinese children repeated low-level red light therapy is effective. But there are unknowns. Still, this is not what's unknown, is the longer efficacy and safety, whether the rebound effects, whether what is the optimum treatment strategies, although these studies are well designed, and how about the underlying mechanism? Now, this is the weakest part. I'm sure the audience can challenge me on what's the mechanism. Then I can tell you I don't know much about it as yet, because it's still in the progress by various groups in the world, right? But then therefore there are further work important. Now this is very prophetic. Double mask and placebo control study was needed, as advocated by Mingguang, and also mechanistic study according to this one. And then this work was also followed up by another group, and again in Shanghai, Professor Xu group. This one is a 12-month parallel group school-based randomized clinical trial in 10 primary schools in Shanghai, another big city, right? Chinese children, right? This time 278 students, and so you see that the incidence of myopia really after 12 months really decreased in relative terms. So again, what's the conclusion is the myopia incidence reduced by 55.6% at 6 months and 33.4% at 12 months. And all in all, the 36.2% reduction in AL. Now, they also examined these children by OCT, and they found no VA structural damage in the intervention. So they're giving some good data on the safety. That was published in 2023, and there are other studies going into more details. For example, no reversible visual function loss that's been found in one study, and combine our LLL, the repeated low red light therapy with OK lens. So that's also been published, and also going to the very detail on the thickness of the choroidal thickness. So all this have been replicated. So all in all, different studies show that repeated low-level RP have strong efficacy, and then And then the intervention group still experience AL shortening after 12 months. So it seems that there's still more to do. And then there's more studies, and this one on 24-month follow-up, right? And there's also different arrangement, the control, and over 2 years, and then they have this 1 year single vision lens, and the other year single lens SSF, and then we start up with RLVL first, and then SVS first, and lastly, you see that the double dose RLLL for over 24 months is most most applicable, right? So we have the conclusion from this study published in 2022, and then there's some information on rebound effect. So there are some modest rebound effects. So overall, with this sort of repeated low-level red light does have some modest rebound effect. But whether or not the rebound effect would be affected, right, is still under investigation, and the rebound from this current study so far did not completely negate earlier results. And there are also collective evidence from other studies that that on the red light wavelength, and then the main difference from meta-analysis all been confirmed from the data so far, right? So, and then there are combination study, and this one particularly, RLL with 0.01% atropine, and found that the light therapy is more effective than 0.1%. That's very low level atropine. And of course, the safety, and this study reported in 108 children on the safety so far is fine, so to speak. There are more studies on the safety, and then further work are needed, but then there's so far no known reversible, irreversible visual function loss or ocular structural changes. So in summary, we gathering from all being reported so far, we could advocate these guidelines to parents and to practitioners, right? On the red light, we get this machine and what to do. So on clinical procedure and practices, all here, clinical safety measures, follow-up, and clinical outcome assessment. So we have been able to advocate these guidelines. In conclusion, we find that this red light therapy much better efficacy in high mouths, right? Are shortening in more half of the treatment group, right? And then the RLL plus ortho-K and enhanced efficacy in poor response to ortho-K monotherapy. Essentially, the usefulness of red light therapy has been established. Although, very importantly, very important for the world. That's why I said it's an emerging field of research that we do need fundamental study, big study, double masking, placebo control. That's also needed. Multi-center and ethnic differences, combine the single therapy, longitudinal, and most of all, mechanistic study. And we know that, including our group, that we are now doing this in animal studies. So I acknowledge, the contribution, the work of my colleagues led by Professor Jason Yam, and the support by Guy and Clement and the team in Hong Kong and in Shantou. And we need collaboration. Hopefully, this presentation draw attention, and you're welcome, to the communication. We really need multi-center trials, and you're all welcome to Hong Kong next year, APAO. So this is Hong Kong, and then hopefully this is attracting you. Thank you very much. Thank you.
Excellent, Dr. Pang. Yes, I think we don't have one speaker. I think going to be two. So the time is adjusted accordingly. So we can have some questions. Time can register.
Dr. Pang, I appreciate the work, the collection, and the various studies which you have shown, and your admittance that we still don't know the mechanism of how these therapies work. But do you have any hypothesis? That's one question. The second one is, you have been talking of reducing the axial length, which is slightly I'm not able to comprehend, because the length of the globe is determined genetically to grow till the age of 18 years. That's what we all know. But I don't think it can shorten. Maybe it will prevent elongation, if that's what you mean to say. Thank you.
Thank you very much. Great question. Very important. And I haven't been able to make myself clear. And of course, a lot of unknown, right? So yes, firstly, on the mechanism, it got to be the stimulating effect of red light, and we know that, in contrast to blue light, which we know that it does not do very well on many ocular cells in culture. That's all been known is red light that could stimulate some growth. But then, of course, you got to have a balance. So our hypothesis so far is still on the activating activity of this 260 nanometer wavelength laser, right? At different dosages, different energy levels in vitro and in vivo. And so there are work going on like that, right, on this essentially on stimulating effect. Then there can be some other hypotheses by other groups. But so far, we're still waiting for publication from different groups, and our data haven't been confirmed yet, so we're still working on it. But definitely, this is an important emerging study, and then we do need more hypotheses. That's why I welcome, collaboration, ideas that we can work together, right? And come back, and then the second question, axial length. Axial length is still the sort of, the axial length that we measure by the IOL master, right, or by the lens star. So that's essentially the axial length that we are talking about, because these are on children coming into our clinic. So that's why we use our clinical equipment to do the measurement, and it should be the group itself. Now, whether or not, as what you said, is a prevention of growth or retarded growth, right now there are possibilities that it could both happen. So, as what I said, we are refining our procedures, right? And then that's right. I cannot be sure yet, can't be sure. But then we are going to confirm by our data. We are following up these children. And actually right now, we have more than 1,000 children undergoing this sort of therapy in our trial. So that's very important, because we do need different groups, right? As we said, and the combination therapy, or even a single therapy cell, the design of the treatment itself would affect us. So we also need to make measurements over a period of 3 years. I think in 3 years we should be able to at least answer partially your question. So thank you very much. Very, very enlightening question. Thank you.
Kelvin, excellent talk. I'm Tokan, Singapore.
Yes.
May I know the amount of red light received by the children compared to, let's say, 2 hours of ambient red light? How does it compare?
It is stronger, slightly stronger, not too strong. The point is, you have this procedure. That's why, the trial itself, we have to do many different of trial, because as what you said, you mentioned very important, it infer to another point, is that these children in this study are exposed to different extent of light in their daily life, right? You are not to shut them up indoor throughout these 24 hours. So that's why you have, in order that you get a real good effort, you need a good sample size. That's one point. Secondly, definitely the amount of light, the energy level that are imposed on these children are slightly higher. So again, we can do a calculation later, right? Because on that, we have to measure, the ambient light, or what's ambient then in this particular season. But then, this study have to be at least 1 year long, right? So overall, we still have to take measures. And of course, the ambient light out, red light in Singapore, in Hong Kong, in India, what happened, under the Indian sun, right? So strong. And also in the north is different. So this is very, very, not just important, very tricky question that, we really have to do a lot of study to verify. And again, I want to encourage our friends to be involved in study of this.
It's very interesting. This study is very interesting, especially the tropical countries like India and other, South Asian countries. Despite we have very good sunlight, it's a paradox that, we are looking for oral type of therapies to look for. I don't know how much is the compliance which is going to happen when the children is looking into the light all the time. will you be able to make them to sit down for some time? So I think we will move on to the next one. We can have a discussion. Yeah, quick question. Yeah, please go ahead. Yeah.
Sorry, Dr. Sirish from Bangalore. I saw that one of the arms of your study has ortho-K also incorporated in the treatment. Am I right about that?
I think
This
it's
is
in
not
here.
our study, but this is reported. Yes, yes.
So
That study
if-
particular, yes,
Yeah.
have a
If that is so, if ortho-K also is one of the arms, it could be a confounding factor when the axial length is measured, that the axial length can go down, at least temporarily, when the ortho-K is done. So is there a comparison between those patients who were exposed to the red light alone versus the ortho-K plus red light, and was the reduction of axial length more in people who had ortho-K additionally? Because that would become a confounding factor. Thank you.
Definitely, you're right, but that's not in that report, right? So that report does not have that in it. So I'm going to drop down this, and our group don't work on ortho-K, or OK lens, right? So we'll arrange, and encourage our collaborators to work on it. So this is very important point on this sort of effect, and I think you have to redesign that study. Based on that study, we have to redesign the protocol, right? And then to observe this effect over ortho-K itself alone, and then ortho-K in combination with red light, and then you can also have to swap, the sequence. That's very, very important in order to answer your question. Your question has to be answered, but not by this report. Right? Thanks very much. Great. Thank you.
I do have a couple of questions, but we will discuss sometime later in the interest of time. Only 20 minutes need to be shared among everybody. So let's move on to the next one. My colleague, Dr. Indumati Mariappan, from LVPI, who is very passionate on the buzzword cell-based therapy. Please, Dr. Indumati. Is this working from that side? It's working? Oh. Oh, there's a different one.
That's
It's the
not
first
mine.
one. You change it to third one. So,
Mic is not on.
it's just coming up. It is changing. It's changing. There's a bug. I think that place there. Show that.
Indumati.
Dr. Indumati.
Is the mic on?
Please.
Am I audible? Mic is not on.
It's working.
It's working?
I'll keep close.
Okay. Good afternoon, everyone. First of all, let me thank the... No, it's not. Am I audible? Yeah. Thanks to the organizers for inviting me and also giving this opportunity. And today I'll talk about cell-based therapy for inherited eye diseases. So we know that cell-based therapies have been explored for ocular surface reconstruction for a while using adult stem cells like limbal stem cell transplantations. Today I'll focus on retina. So what do we do for some of the retinal degenerative conditions? So we know that in the retina there are multiple layers. The neuroretina, there's 3 different layers. Most of the photoreceptors lie in the outermost layer. This is the layer that is degenerating in many of these patients. Another layer that degenerates is the RP cells, which supports the photoreceptors. So in cell-based therapy or for gene therapy, the cell type that is being targeted are either RP cells or the photoreceptors. So when you look at the genes that are involved, because this is inherited conditions, can be age-related as well, but inherited conditions, over 300 different gene mutations have been implicated in the disease. And when you look at individual gene mutations, it's very rare, so it comes under the rare and orphan disease category. And so far, for the gene therapy, we have one product which is commercially available, approved by FDA, which is Luxturna. Many of you are already familiar with. And we also know the cost implications too. And most importantly, this is gene mutation dependent. So in case of other diseases, we really have to develop individual vectors for each of these genes. That's a workload that is ahead of us. And alternatively, we can also think about developing a gene agnostic approach wherein you can transplant the cells that are degenerating, whether it's RP cells or the photoreceptors can be transplanted. So many allogenic iPSC or ESC derived cells have been currently been explored in several trials, and outcomes have also been reported by several of the groups in UK, US and Japan. And so far what we know is these cells, CS or iPSC derived cells, at least in terms of safety, we are more convinced. And with respect to efficacy, we need more trials to teach us about what is the long-term outcome of this. So in addition to that, we have gene editing coming up. So one approved trial, first trial for AIOS2025, which is LCA10, is also been reported. But more has been explored. This is for in vivo, directly delivering the gene editing construct into the eye and looking at the outcomes of these patients. But alternatively, you can also combine cell therapy with gene editing by using patient-specific stem cells and editing them in the dish outside the body and making normal cells as a cell-based therapy. So today I'll give a couple of examples of our effort in this direction. So ESCs have been extensively explored. So in the recent past, iPSCs have gained a lot of importance, and it's been very practical to generate patient-specific iPSCs by taking skin biopsies or blood samples from the patients and reprogramming them to become pluripotent stem cells. And once you reach this stage, this can be differentiated into any cell types of the body. In this case, we are interested to develop retinal cells. Either RP cells, which are enriched populations, can be derived from the differentiated populations, or you can generate the entire neuroretina as retinal organoids. So either of these can be transplanted into the patients for therapeutic applications, and many trials are currently ongoing. So how good are these cells? Are we fully mature enough and functional to consider for clinical applications? So here is a cross-section of the normal retina stained with rhodopsin, which is the photoreceptor specific marker. So when you look at the organoids that are generated, you get about, in about 3 to 4 months time in culture, you get a good organoids which are about 3 to 4 mm in diameter. And when you make a linear sheet, you can easily make about 7 to 10 mm long retinal whole thickness retinal graft. And if you mature it to about 4 months, you get nice photoreceptors, which are rhodopsin stained, right? Just like the organization in the native retina, the outermost layer is lined with photoreceptors which are fully mature. So not just rods, but also cones. Cones are very important for macular because your fovea is enriched with cones. For all bright light vision, you can get beautiful cone-enriched organoids as well. And these organoids there are trials where they're exploring directly transplanting such organoids into the patients, or use them to purify and enrich only the photoreceptor cells and use them for transplantations. And coming to so that's what I was meant. There's a circular graft. When you linearize, you get a graft about 6 to 8 mm graft from each organoids, which can be transplanted into the patients. From a single dish, you can, starting from 1 million, you can easily get about 30 to 50 organoids in one culture. So that's the kind of scale up that's possible. It's not really scale up. Even for a small scale preparation, that's feasible at this point. We have the technology right now. For RP, again, you can make enriched populations of highly pigmented, fully mature RP cells which are functional. You can prepare them as a sheet on a graft, like a collagen graft or PLGA scaffold, make a sheet, or you can prepare them as suspension and can be injected subretinally. So that's totally feasible now. So that's what I meant. So you can make sheets or suspension, or you can have the retinal organoids for photoreceptor transplantation can be injected subretinally. So that's what the trials, there are many trials that are ongoing. So first the first in human trial in India started just last year. In collaboration with a company called iStem Private Limited, the trial has just finished recruiting the first phase of 9 patients with 3 different doses. It is a multi-centric trial. One of the centers is our own centers, which is Helvi Prasad Eye Institute, Hyderabad. The other centers at Delhi and Maharashtra. So the patients have been, recruitment has been already completed and they are being followed up. So what is known from the experience, clinical experience, is at least over the 6 to 8 months follow-up of the first few patients, we know that it's well tolerated. And it's important to note that this is an allergenic product in the sense that patients have to be immune suppressed 1 week, primed for immune suppression 1 week prior, and continued up to 12 weeks. That's the protocol. So this is for AMD, GAA secondary to AMD. That means these are older patients more than 60 years and they are already immune compromised. You really have to put them on immune suppression for the cells to survive there, because you don't know already there is an inflammatory conditions going on. So this is a major point, because though allogeneic cell therapy can be gene agnostic, irrespective of the genetic mutations, you can really recruit patients. If it is RP transplantation, as long as the photoreceptor, there are some residual photoreceptors are there, the transplanted RP cells should be able to support and prevent the further degeneration of the photoreceptors. That's the idea here. But then immune suppression have to be a case, but after 12 weeks, if you withdraw immune suppression, what is the fate of these cells is something that we will have to know, wait and watch. So now thinking about this, so can we develop an autologous approach? That's another thing. So if we, now that IPCs can be derived from many patients, can we make autologous in the sense, edit the mutations, provided we know what is the gene involved and the mutations can be corrected. So that's the approach. So people are looking at both ex vivo editing and also in vivo, in the sense you can deliver this CRISPRs. People use editing system. We have multiple editors now currently available. The advanced editors are really very good in the sense that they don't even cut the genome. So without cutting the genome, you can make modifications, specifically point mutations can be corrected. So now these constructs can be packed into viruses. Well known and the very known virus type is the AAV. It can be packed into AAV and can be delivered. Alternatively, as I was telling, we can combine cell therapy with editing by taking patient-specific iPSCs and edit it in ex vivo in the dish. Once the editing is confirmed and corrected, and you can make the retinal cells which are tailored for the patient, in the sense it can be an autologous therapy. So we took this approach to establish a proof of principle. I'll give you 2 examples today. So we use an advanced high-efficient editor, which is a F1 Cas9, not the traditional Cas9, which is well studied from Streptococcus pyogenes. This editor is highly efficient in the terms of off-target recognition, and it doesn't cut elsewhere, even if there is a single point mutation base difference between the target site and the off-target site. So using this editor, we collaborated with the group at other collaborators who have made an AB version of this Cas protein, which converts adenine base by deaminating it. So once the adenine is deaminated, it becomes inosine. When the polymerase reads, during the repair process, you get a guanosine. So you can do an A to G conversion. So we tested this in 2 patient models, which is LCA2 in a gene called RP65 and LCA12 in RD3. So this is the So this is a fundus image of the patient, and it has a mutation in exon 9. So the stop codon, we are trying to convert this A back to the tryptophan residue. So you can see that in the patient-specific edit, so we could identify 2 clones where both the alleles, because it's a recessive mutation, both the alleles got the mutation, and you can completely correct this to get 100% edit. And if you differentiate this line into RP cells, the patient line doesn't express the protein RP65. After correction, you can completely restore the expression. Now this cell is edited, patient's own cells, corrected for the normal protein expression, can be transplanted to the patient as an autologous approach without the need for any immune suppression. And coming to the next example is congenital case again, LCA12. This gene is important for photoreceptor function in the sense that, again, it is a splice site mutation. Because the intron is included, there is a premature stop codon, therefore a truncated protein is made. So here we are trying to restore normal splicing by editing and correcting this. So patient-specific line, again we edited. In this case, we have edited, and this edit is an heterozygous edit, in the sense only one allele got edited, but then because it's a recessive condition, even if one allele is functional, it should partially restore the normal function of the protein. So since it is a photoreceptor specific gene, we need to make retinal organoids. We made the retinal organoids and looked for the restoration of normal splicing. So if you see a normal splicing happens, you get a small product in an RT-PCR, whereas if the intron is present, if there is an abnormal splicing, then you have a bigger product. So we validated this. You can see that in the patient line, the normal product is totally missing, whereas in the edited line, this is completely partially restored because one of the allele is corrected. The other allele is still a mutant allele, but still they should restore the function of the protein. So now we know that the ex vivo editing can be an option for patients where the cells are already gone. In vivo, we cannot do anything, so we can take the cells out, make an iPSCs, and you can edit and correct. But so you can use allogenic as a gene agnostic approach, autologous for patients who cannot tolerate immune suppression. So both options are now possible. For in case of patients where the cells are still preserved and you can go for gene therapy vectors are available, then you have in vivo gene editing using either gene therapy or gene editing. So these are possibilities that are available in front of us. So many trials are still going on, and hopefully we'll learn more about the feasibility and the cost effectiveness and affordability of these therapies in the days to come. And this is a team that has worked with us, basic research team and the entire clinical research groups and the funding agencies for the support. With that, I'll stop and I'll be happy to take any questions.
Thanks, Dr. Indumati. Any quick question? Can you just, is there any intermediate efficacy you got out of eyesight study?
Out of which?
Eyesight, the trial.
Eyesight, yeah. So no, not yet. They haven't reported it. But what we know internally from our group, from the patient that has gone in at LV Prasad, so the cells have been well tolerated. There's no any systemic adverse events reported.
So you're looking into more the safety.
Safety, because this is a phase 1-2 study. So looking more safety, we are pretty confident across the different patients. Efficacy need to be looked at. So, but we are looking forward for the combined outcome of the whole case series. Yeah.
Very good. I think we are also eagerly looking forward for the outcome of the study.
Yes.
There's no question. Shall we move on to the next one? Thank you very much,
Okay,
Dr.
thank you.
Indumati. It's excellent work.
Thank you very much. Great work. So we welcome our next speaker, Professor Kimaram Cahill. Professor Kumar is a pioneer of eye genetics in Asia, and he and his team published Nature genetic papers back in the 80s already, sometime back. Of course, he's always young. So Professor Kumar will share with us his work on translation of genomic knowledge in ophthalmic clinical practice. Professor Kumar, please.
Can I have my first slide? I can't see my slide on here in my laptop.
No slide is the other side.
Should I be on this side or the other side? Sorry?
It's on that desk?
Can't see here either. I'll open it myself. I hope this will work on the screen, main screen. Okay. Something. I'm getting it here, but I don't know if this is the one over there. No, this isn't on the screen actually. नहीं भैया ये चेंज करते हैं उधर नहीं आते हैं Sorry, this is still being fixed.
सर सही तो है
If you change it, you will
अच्छा उधर करना है आपको
उधर नहीं होते हैं चेंज करना ना
अब आ रहा है ठीक है
Okay, thank you. And at the outset, I'd like to thank the chairs as well as the organizers for giving me this opportunity. So I'm going to talk on the translation of genomic knowledge in ophthalmic practice. At the outset, I have to agree that I'm not an ophthalmologist, but more a vision scientist and a geneticist. So what I'm going to talk about is on this slide, and I'll start with the paradigm shift in clinical practice. So basically in the 19th century, clinical medicine became more reinforced, and it was based on signs, symptoms, diagnosis and management. In the last century, it moved into preventive medicine, where you have target groups which you can screen clinically or biochemically, and then catch them early so that you prevent the disease. But in today's world, in this century, the field is more personalized and predictive medicine. So where you do deep phenotyping and you do all kinds of omics markers. For example, if I'm going to develop AMD in 5 years from now, so as a patient, I say that 5 years later, I can't read properly, and then I go to the ophthalmologist and they diagnose that I have AMD, but that it's too late to reverse the disease. So it's today that whatever molecular changes that is happening in my retina, in my macula, has to be picked up either in the tears or in the aqueous humor or in the serum, so that I'm going to develop AMD 5 years later. Okay, and then you start to apply all kinds of preventive methods. So that's predictive medicine, and that's today possible because of genomics and various other omics that is available. So this is a central dogma of biology, where DNA finally becomes protein and then becomes an organ and an individual. But this central dogma is also becoming a central dogma of medical practice today. So at every stage, from the genome till the phenome, or the physical appearance and characteristics, we can actually intervene by various methods and block it, okay, or enhance it, whatever that you can manipulate, and either reduce the gene expression or you can actually enhance it. And therefore you can reduce the morbidity and ultimately the mortality. And that is the fundamental basis of my talk today. Now, how we apply genomic knowledge in patient care as a geneticist and a genetic counselor. This is a patient with oculocutaneous albinism. Because of time, I have to rush through and cannot explain too many things. But the point is that this is the individual who has oculocutaneous albinism, has reading disabilities, but she's a graduate. And she came across an individual with beta thalassemia. Okay. So they got married. Now the question is whether the next generation will get either one of the disease. Okay, I've been doing genetic counseling last three to 3.5 decades, and mostly patients come with one particular genetic disorder. But today the scenario is changing, like the Western world, where both sides you have a genetic problem and they want to know whether it will affect the next generation. So to cut the long story short, we did genetic investigations. We knew that this lady had OCA2 gene variant. One was likely pathogenic, another one uncertain, but we are sure that is also causative. And her partner had a HBB variant, which is indicative of beta thalassemia trait. So we did what is called a carrier screening, and this is too much of information, I'm not going into it. But ultimately what happened is that with all that genomic screening that we did, or genome screening we did, we came to the conclusion that there is a 50% chance of recurrence of beta thalassemia trait in the next generation, and there is nil chance of OCA recurring. Okay, now that's the power of genomics and predictive medicine, personalized medicine today. And so we practice in many, many instances. I'm not going to go into too many of cases. Let me move on to the next part of my talk. How will the prescription practice undergo paradigm shift? There are many, many things happening. And in fact, we had a talk about stem cell. So there are gene-specific therapies, and there are 300 genes. There are too many of them. It's not an economic model to treat each one of them because we have few hundred people with certain genes, and therefore, no company is interested. But agnostic therapy is sort of picking a bit of attraction now because it is non-gene dependent therapy. So the mutation directly doesn't cause the retinal degeneration. Okay, so it's inflammation, apoptosis, immune response, oxidative stress, many mechanisms that ultimately destroys the photoreceptors. Okay, so we can block all of them. And the biggest advantage is that it's almost similar, the methodology is similar to gene therapy, but then this is non-gene dependent. And therefore you can have an anti-inflammatory molecule, TNF alpha, or, antagonistic that you can prepare and put it in the eye. And the biggest advantage doing that is that it cannot not only apply for Mendelian diseases, but also for complex disorders. So you have malignancy or you have autoimmune disease. Most of the mechanism starts with inflammation, and therefore these kind of companies can survive very well and make profit simply because they can play on both sides of genetic disorders, on Mendelian as well as complex. So again, there is a battery of mechanisms by which you can block the protein synthesis with siRNA, mRNA, small molecules, oligonucleotides, cell-penetrating proteins. These are all not too much of up in the air story, but this is actually true. I'll show you why. So this is a nice paper about how cell-penetrating peptides can be injected into the eye, similar to gene therapy. And that's a list of ocular delivery agents of cell-penetrating peptides. I know it's a busy slide, but all that is going either in animal studies or in preclinical studies or in clinical trials right now. Okay, anybody who wants more information, I can give you this, but I'm not working in this area. So this is actually a pharmacological potential of small molecules for treating corneal neovascularization. Okay, so you can see a big list of them. There are many, many molecules, and some of the molecules' mechanism of action is not known, but still, Dr. Vel Pandian will know this better than me. But all this is coming true. So therefore, you have gene-specific approaches for retinal degeneration, gene-agnostic approaches. But gene-agnostic approaches, you have to start pretty early. You can't go at a very late stage. If it's a late stage, stem cell therapy is the best, or any kind of cell therapy which is, again, agnostic in nature. So they're also trying to revive, embryological genes so that you have some kind of pseudo photoreceptors, but it has to be again started early. And again, I can give you this reference. Anybody is interested on. Oligonucleotides are coming up in a very, very big way. So the DNA, the biggest advantage is that they're complementary in nature. So you put a small DNA, it'll go bind to the genomic DNA and block its activity. So there's not only ophthalmic, but there are many other fields, and all these are available across the counter now. Okay, it's not something that is sitting in clinical trials. So small molecules are basically where you get very small fat molecule, and you can put anything into it, siRNA, peptide, or a small DNA, microRNA, and so on. So therefore, you can inject them into whichever cell or tissue that you're interested in, and then they can simply act and produce less morbidity or inhibit the deleterious mechanisms that are happening in any part of your eye. Okay. So these are exosomes. Again, as I mentioned, there are quite a lot of them going on in research, again in ophthalmology, besides other fields of medicine. So with that, I come to the last part of my talk. So what is the future now? Okay, where are we going towards? So, like doing any other investigation in your clinic, it is mandatory that you have to screen the genes. Okay, of course, there is a need, you have to do it. There's no way that you can have a child with a genetic disorder and you don't do a genomic screening. So this slide tells you that this is a traditional care pathways. A child comes with congenital cataract. What people did for the last century is that they will sit and wait and watch what are the symptoms that are appearing, signs and symptoms in the child, and they'll make a diagnosis, syndromic or whatever. It'll take 3 years, okay, but all damages are done by then. But today the child comes in with congenital cataract, so you screen with next-gen sequencing, and then you know what the gene is, okay, and then you start to see what signs and symptoms will appear. Accordingly, you can sort of counsel the parents, and they can go for the appropriate rehabilitation services and help the family and the child to recover faster, okay, and then integrate into the society. Okay, so therefore I'm come to the last slide summary, and so I spoke about the paradigm shift. We are in the era of personalized and predictive medicine, and we do genetic counseling using genomic screening, and then there are lots and lots of changes happening, and the way you clinicians are going to prescribe medicine is going to change a lot, and I'm sure Professor Will Pandian will be able to answer the many questions that you might ask after I finish my talk. So the future is you have to do a genome screening. So as a clinician, you suspect a bit of genetics or age-related disease, it's AMD or diabetic retinopathy or any kind of degenerative disease, you have to screen the genome. Okay. With that, I come to the last slide of mine. And so I work for Narayan Nethralaya, so there's a clinicians group. You can see Dr. K. Bhujang Sethi as well as Dr. Roy Sethi here, and this is Professor Shomi Bhattacharya. This is a research group, and Dr. Arka Ghosh, Debashis Das manage that part of the Narayan Nethralaya. These are my students in Chennai, PhD students who I guide, and they help to get this talk done. So I'm thanking them. With that, I finish my talk. Thank you.
Thank you very much, Professor Kumar. Time for a quick question.
I have questions.
If you ask question, Professor Valpandi, you have to answer it.
We'll talk it over
Okay, go
coffee.
ahead, yeah. Yes, please. Question. Yes, madam. In India, we have no other choice. The costs are coming down, and it's sort of most of the screening that you do now is under 20,000 rupees. 15 to 20,000 rupees. And mostly the patients come for premarital counseling, and then preconceptional, and then prenatal. For these reasons they come, and then you screen them, or it could be for clinical diagnosis where they missed or they suspect a syndrome, where all you can help. But in India we have to pay from the pocket, and many parts of the US also, not that every state gives you coverage for genomic screening. Okay. Yes, again, having finished my talk, genomic screening is not as easy as I told. There are many issues that we are facing, particularly one called as variant of unknown significance, and this is a nightmare in the field, where a particular change we don't know is causal of the disease or not. But then there are many other ways by which you can confirm it. So therefore, for example, in my particularly retinal degenerative diseases, your hit rate is pretty high. Unlike a few, like for example, neurodegenerative diseases, childhood related, the hit rates are very low. Yeah, so comparatively.
All right. Thanks very much. I think we have to move on. Thank you, Kumar.
Thank you.
We can have more discussion later. Thank you.
Thank you.
So we welcome our next speaker, Dr. Shenzhen Wang from Singapore, to share with us, very interesting work that he's conducting in the series Singapore. Thank you, Dr. Wang.
So can anyone hear me? Okay, so I thank the organizers for inviting me here. I'm a basic scientist. I work on exfoliation syndrome. And essentially, exfoliation syndrome is defined by the presence of this white specks in the eye. We are trying to find out how this arises and how do we control them. I have no conflict of interest. When we talk about exfoliation syndrome, LOXL1 is the most important gene. We know that common variants are associated with protection, but sometimes the effect is flipped. And rare variants, my collaborators did a survey a long time ago and found that they are enriched in controls, which means that they're associated with protection. But somehow it's also paradoxical because LOXL1 protein itself is a constituent of exfoliative deposits. And LOXL1 expression in the anterior chamber is increased in early disease, but decreases in advanced disease. And the second gene I'm talking about is really CYP29A1, which is a metabolic gene that we discovered to be associated with exfoliation syndrome a few years ago. Essentially, if you are an individual who carries a loss of function allele for CYP29A1, you have about twofold increased chance of getting the disease. Importantly, people with this particular mutation in the Japanese also have an increased risk of progression to blindness. The most important thing to take note for this talk is that CYP29A1 expression is downregulated in patients, even if they don't carry CYP29A1 mutations. So that's a key point. And the next question we ask, since we have both the presence of protective LOXL1 mutations and deleterious CYP29A1 mutations, is do we have people who carry both? And if they do carry both, what is their phenotype? So we looked at our patients, our cohorts, and essentially we found 4 such individuals who carry both protective LOXL1 mutations and loss of function CYP29A1 alleles, and all of them do not have the disease. So it appears that LOXL1 mutation overrides the risk imparted by CYP29A1 mutations, and that was our impetus for starting this study to look at both CYP and LOX in the same cell. So to do this, so essentially these are the alleles. We use RPE cells simply because we can't find any ciliary pigmented epithelial cells, but we know that they're continuous in the anterior chamber, and they also express high CYP expression in both the RPE and the ciliary epithelium. So the important thing we know is also that they express LOXL1 endogenously. As you can see here in the cell lysates, you see that there is you can detect LOXL1 in the cell lysates. Most importantly for this talk is that you can take the spent media and you can do a fractionation. You can detect the soluble LOXL1 in the media, and then if you do a very high G ultracentrifugation, you can also find insoluble LOXL1 in the media as well. So if you look at, and then if you express LOXL1 as you would in early disease where LOXL1 expression goes up, you see that there's an increase in LOXL1 abundance in all fractions. So if you do IF, you will find that LOXL1 does not colocalize with intracellular markers. It actually colocalizes very strongly with fibronectin, which is the ECM, the extracellular matrix. So how do we model loss of CYP29A1 expression? So essentially, this is the pathway here. If you have loss of CYP29A1 function, you're going to have an increase in 24-hydroxycholesterol because that's a substrate. And we also know that 24-hydroxycholesterol is a ligand, or because it's oxysterol, of LXR, which is a very, very well-characterized nuclear receptor. And of course, we do have inhibitors that inhibit LXR. So what we did was simply take LOXL1 overexpressing cells of RP cells. We treat them for 48 hours. We don't see much happening in the lysates, but you see 24-AC treatment causes LOXL1 to dissociate from the fibronectin fibrils, and they appear to be more diffuse. If we look at the culture media, which is essentially extracellular space, see that there's a strong increase in both soluble and insoluble LOXL1 in the cell culture media. And this is caused by LXR activation, because if you then co-treat with GSK2033, you can reverse the effects. So an important thing that LXR does, very well known, it causes cholesterol to move out of the cells in a process called cholesterol efflux. So we ask the next question, which is, can the loss of cholesterol itself be sufficient to cause LOXL1 to be released from the ECM into the cell culture media? And to do this, we simply strip cells with MBCD, as you can see here. Once you have MBCD treatment of cells, you simply replicate what you see with 24AC. Loss of cholesterol, again, leads to an increase in the LOXL1 soluble fractions in both in cell culture media, and you can also see that this localization pattern resembles 24AC. So here, we essentially show that increase in 24-hydroxy cholesterol leads to LXR activation, and LXR activation causes cholesterol to leave the cells, and this essentially causes LOXL1 to be removed and goes into the cell culture media. Now, this is all well and good. This is in cells. Can we see in the patients? That's the most important part. So essentially, we looked at markers of LXR activation in aqueous humor simply because they are most correct approximation for extracellular space, because they are essentially extracellular fluid. And then we're very lucky to get a lot of aqueous samples from Dr. Ozaki, who's sitting there. Essentially, we can use mass spec to quantify the amount of esterified cholesterol species in the aqueous humor of patients. And as you can see here, there's a very strong and significant increase, especially in patients who have late-stage exfoliation glaucoma, when glaucoma is already observed in their eyes. And so this tells us that at least in this cohort of patients, LXR is activated. And it's most likely because of loss of CYP29A1 activity, either due to mutation or decrease in expression. So can we model this in our cells? So we can, because we can now express CYP46, the upstream enzyme, and we can see that essentially 24-OHC levels go up, and you can see less free cholesterol in the cells and more esterified cholesterol in the cell media. Can we now, then we looked at these cells in the chronic context. We note that there is a very new inhibitor of CYP46A1 activity called stocostat. So when we repeat our analysis, we looked at essentially the amounts of LOXL1 in the cell media. We see that CYP46A1 cells, they also have more LOXL1 in the cell media in both the soluble and the insoluble LOXL1 fractions. And if you now treat with either stocostat or GSK2033, you can reverse the effects. So there's some kind of a therapeutic angle that we're developing with regards to exfoliation syndrome. Now we also know that this release of LOXL1 away from the ECM could be pathological. So we can predict that in patient aqueous humor, you should find more LOXL1 protein, and they will be more abundant. So this was really what we did. We had a few batches, and we're doing it for multiple batches now. You could see that in especially exfoliation glaucoma patients, you have much more LOXL1 in the aqueous humor compared to controls. So this is all well and good. But what about the LOXL1 variants that I just mentioned were protective? So what is the basis of the protection? So what we did essentially was to make individual cell lines that express these LOXL1 variants. The red ones essentially are protective, they are enriched in controls. Black ones are wild type, common variants, and rare variants that are not associated with protection. And you can see they somehow have less LOXL1 being released into the extracellular space, as you can see in both the soluble LOXL1 and the insoluble LOXL1 fractions. So essentially, protection from exfoliation syndrome comes from these proteins not being able to be released in the extracellular space. This is what our hypothesis is now. So what's the basis of this lack of release. The basis of lack of release, in short, is there is simply less stable in the cells. If you do pulse trace experiments, you can see that wild type and P31 which is not associated with protection, LOXL1 is essentially very stable. But then if you check the protective variants, they're extremely unstable, and this is caused by them being constitutively targeted for degradation, because you can rescue the phenotype by just inhibiting either proteasomes or autophagy. So essentially, this is essentially what our working model of exfoliation is right now. So normally, LOXL1 in the eye, in the ciliary epithelium, you see that it's nicely in the extracellular matrix of the ciliary epithelial cells. Then, when there's a decrease in CYP39A1 activity or increase in LOXL1 expression, there's more 24-hydroxycholesterol. 24-hydroxycholesterol is a mobile metabolite. It moves. It crosses cell membranes, goes into the non-pigmented epithelium, ciliary epithelium, activates both LXR, causes cholesterol to be released into the extracellular space, which is why you can see more saturated cholesterol in the aqueous humor. Then subsequently, you get LOXL1 being released from the ECM into the into the extracellular space as well, and that's why you can see more LOXL1 in the aqueous humor as well. So I've come to the end of my talk. This is the people working on it. I'd like to thank Dr. Ozaki, who provided most of the aqueous humor samples, and I'm happy to take any questions. Thank you.
Thank you very much. Questions from the floor? Any questions? So what's the clinical implication of this mechanism involving 24-hydroxycholesterol, Shen-Shu?
So essentially, the reason why you have more 24-hydroxycholesterol is because there's an imbalance, there's too much CYP46 activity. So one way is to use an inhibitor of CYP46 to decrease the levels of 24-HC so that you don't get the downstream effects that you see in patients. The other thing would be to, I didn't show this, but LXR inhibitors work as well. Also, ER stress. If you can use chemical chaperones to make LOXL1 fold properly, you can also alleviate the release into extracellular space. So these are the three approaches that we're currently exploring right now. Yeah.
Right. So you are moving on to prove it. Yeah?
Yeah.
Question? Speak to the mic, please. Here.
What's happening to the secreted LOXL1 in these patients? So there's too much secretion, so are they getting aggregated, or what's happening in the ECM?
So that's a very good question. We speculate that the insoluble LOXL1 fraction will serve as a seed for other proteins to bind, and that's how they cause exfoliative deposits. Yeah. So that's the reason why if you have LOXL1 normally in the ECM, nothing happens, but when it gets released, bad things happen. Additionally, there's a second clinical feature, which is aqueous flare. The blood aqueous barrier becomes compromised. We think it's probably because of this LOXL1 being released. It's not in the ECM where it enforces the barrier. It's now mislocalized, and that's the reason why you get this phenotype.
Yeah, that's great. So I just have one question about this 24 hydroxycholesterol. Is this what is it acting through a receptor, and what type of pathway it's going out to it?
So 24-hydroxycholesterol is oxysterol. So normally it's a signal for the cells that there's too much cholesterol. So when there's too much cholesterol, it goes and binds the nuclear receptor LXR, liver X receptor. LXR is the effector. Yeah.
Thank you. Now, before thank you, Zheng, right? Fantastic. Well, we'll have more discussion later. Thank you very much. So before the last talk by our last speaker, Professor Shubha Simva Prasad, in recognition of her great contribution in the field and to India and to the world, we have a presentation. Please come. I have the privilege to present it to her. Thank you. Please come. Sorry.
Now we invite Dr. Shobha from Mophils. Along with his recognition, she's going to talk on retinal non-perfusion and macular ischemias. Please, madam.
Good afternoon, everybody. Sorry I was late this afternoon. So my topic is on retinal non-perfusion, another emerging area of research. So these are my disclosures. So, so far, our understanding on retinal non-perfusion is based on retinal imaging. So we moved on from color photographs, 7-fuse to ultra-wide field, then to angiogram, and then OCT-A, OCT, and very recently AIOS2025 as well. Are we there yet? Not. So I think it is really an emerging field. So based on color photograph, if you see these features, clearly we will estimate, at least in our heads, that this patient has retinal non-perfusion. But color photograph by itself cannot assess retinal non-perfusion. And we all know from all the DRSS studies that the higher the DRSS score, the more likely the patient is going to have increasing retinal non-perfusion. And then recently, Paula, Silva, etc. have published quite significantly on the predominantly peripheral lesions on ultra-wide field imaging, again suggesting that they are a sign of ischemia. So if patients have PPL, they have 4.7-fold increased risk of developing PDR because it is a surrogate for peripheral ischemia. Angiogram, this work starts published in 1981, so 25 years ago. The Japanese group already knew that mid-peripheral retina is more prone for retinal non-perfusion than the posterior retina. So they are the first to get involved. But retinal non-perfusion progresses towards the center. So it is more pronounced. The more retinal non-perfusion, the more likely the patient is going to develop NVI and NVD. So in order of retinal non-perfusion, degree of retinal non-perfusion, you may get NVE, progress to NVD, then to NVI and NVG. So we did some work on, because we have ultra-wide field, we tried to look at all the retinal non-perfusion and correlate it to the diabetic retinopathy severity, and we find that if they have peripheral non-perfusion, they are likely to develop NVE. But if the non-perfusion is more in the center, or it progresses to the center, those patients are likely to have NVD. So that's a generalized sign of retinal non-perfusion. So it's almost like retinal non-perfusion phenotypes. Topographically, we know like peripheral non-perfusion is defined as more than 6 diameters from the optic disc, and then mid-peripheral is 6 to 4, and then central is 4 disc diameters around the posterior pole. But generalized is all through. And the peripheral one is the slowest to progress and is the least common. The mid-peripheral is the most common and the fastest to progress to NV. And the generalized one suggests that the patient is likely to be due to poor control of diabetes and likely to develop NVD as well. So we also looked at the total retinal non-perfusion that is likely in a patient with PDR. And this accuracy test suggests that we need about 100 disc areas of retinal non-perfusion for an eye to develop new vessel disc. This is the DRCR protocol AA. They produce the same results, but of course in a larger population and in in a trial. And they also showed cumulative proportion of disease worsening or progression to PDR is more in eyes with highest non-perfusion. You can see the Kaplan-Meier graph. So at this point, we have to credit Ramin Tadayoni because he tried to do capillary non-perfusion work on OCTA. And he found a mismatch between retinal non-perfusion on OCTA versus the presence of PDR. And he suggested that there is significant overlap of non-perfusion index. Non-perfusion index is defined as the total area of non-perfusion divided by the total gradable retina. And he found that there is significant overlap between NPDR, severe NPDR eyes and PDR eyes. So, and the reason for this disparity, he suggested, was probably because there is more peripheral non-perfusion that is not measured in OCTA. So it's actually a device problem rather than our understanding of the knowledge. And we are hoping this EVIRET study, the followers of Tadayoni group will come up with many more papers because they are the ones with the largest collection of OCTA images in studying retinal non-perfusion. This is the quantitative metrics of fluorescent of capillary non-perfusion. So this is Justice Eller's work. Previously, we only said, as we did, we just say central non-perfusion, peripheral, combined, generalized. But now they are able with AI, they're able to measure the ischemia, give quantitative measurement, and also do microaneurysm turnover and leakage. So leakage is quite new for us in diabetic retinopathy. Peripheral retina, we have always seen leakage, but never put a figure onto that. And Justice Eller's work suggests that peripheral leakage around the retina is as strong as indicator as capillary non-perfusion. So now we have 3 markers of capillary non-perfusion, i.e. ischemia or NPI, or count the total area, microaneurysm turnover, and now NPL or leakage. Now we know that ranibizumab and many other anti-VEGF agents improve the DRSS score in about 30 to 50% of patients. But this is the Clarity trial we did. Despite all the improvement we see, cosmetic improvement, all the new vessels disappear, PDR patients become NPDR, all this magical treatment, the non-perfusion doesn't change with anti-VEGF. So actually, if you see very carefully, the non-perfusion actually increases as the disease progresses. So anti-VEGF have no effect on the non-perfusion. And also, we know with the DRCR protocol S suggests that even when patients are on ranibizumab, there is visual field loss, which means the capillary non-perfusion is still progressing, causing the field loss. Usually we, before the era of ranibizumab anti-VEGF agents, we had PRP only. So we attributed all field loss to PRP. But this study was really good in that it showed us that whether you treat the ranibizumab or you censor the patients who had PRP, they still develop visual field loss as a result of retinal non-perfusion. So the disease progresses. Next few minutes, I will describe foveal avascular zone and macular ischemia. So we all, from the days of fluorescent angiogram, we all know the ETDRS paper 10 that showed us how to grade diabetic macular ischemia. So if there is more than half the foveal avascular zone that is enlarged and distorted, we know that we call them foveal diabetic macular ischemia. But we also know that we see a lot of patients with enlarged, irregular foveal avascular zone, but the visual acuity remains normal, right? We have seen all these patients. So this is Amani Fauzi's work. So she showed that actually it's a deep capillary plexus perivenular area of non-perfusion is the first sign of capillary non-perfusion, even before the patient develops the first microaneurysm in the retina. So actually the drop-off is quite early, and therefore if you're to do preventive treatment, it has to be really, really early. I know we can't do anti-VEGF or intravitreal injections as preventive therapy, but if you have drugs, oral drugs or topical drugs, maybe this is the time you have to start the treatment. This is some of our work where we're trying to double on OCTA as well, and we clearly show that with increasing severity of diabetic retinopathy, we see higher standard deviations of capillary non-perfusion, both in the deep and the superficial layer, more so in the deep. Now, we did a follow-up study, so 12-month study on patients with natural history diabetic macular edema in PRP-treated PDR eyes. We just wanted to see how the foveal avascular zone changes over 12 months. And you can see here we had 4 visits over the 12 months. There was no change in any of the OCTA features in the 12 months. Okay, so the true worsening of foveal avascular zone beyond the margin of measurement error was less than 10%. So this is very tricky, because if you are going to do a trial to look at changes in foveal avascular zone, it can't be a small trial, right? And a short trial, because at 52 weeks, nothing happens even if no treatment is given. We also looked at all the changes in visual function and vascular metrics over the 12 months. We had several parameters, visual function as well as structural changes, and actually all of them showed deterioration, but the only significant one was still for foveal avascular area. So despite all the new terminologies we have learned about superficial deep vascular plexus, their density, their tortuosity, fractal analysis, we still end up saying foveal avascular zone enlargement is actually the best metric to look for in DMI patients. But despite this enlargement, we saw no visual function changes, no BCVA change, no contrast sensitivity change, no microperimetry change. So clearly, any clinical trial in this area should run more than 12 months. So there are OCT phenotypes in diabetic macular edema. So if you have no drill and no EZ loss as the reference, that means they're the good eyes with enlarged foveal avascular zone. If you have drill alone with no EZ loss, the patient is going to lose at least 1 line of vision compared to normal, compared to those with enlarged FAS. If EZ loss alone is considered, they only lose very little vision. But if drill and EZ loss are there, then they lose 2 lines of vision. So really, the structural changes drive visual acuity. So this is the anti-SEMA3A pathway that is being experimented now with Boehringer Ingelheim, looking at whether this axonal guidance molecule, that is, it inhibits endothelial cell addition, and therefore it can grow into areas of capillary non-perfusion to try and reperfuse that area. That is the fundamental mechanism of action. There have been the Hornbill trial that used this on previously treated patients with PRP but had enlarged FAS. They seem to think that there is some minimal action there. They saw a few vessels in the foveal avascular margin. They believe that is a good sign, but for me, it may be even an OCTA repeat that shows a new vessel that appeared and disappeared, because foveal avascular zone on OCTA is really tough. And now they're running the Crimson study, which is patients with moderately severe to severe non-proliferative diabetic retinopathy with enlarged FAS, and giving these as intravitreal injection of semaphorine 3A to see whether the foveal avascular zone can be modified. So there's several unanswered questions in this field. For example, we don't know the threshold of retinal non-perfusion required for neuronal damage. We also don't know why the discrepancy between DRSS and capillary non-perfusion. Probably it may be due to the anti-VEGF just working on the leukostasis part, but no effect on anti-VEGF and non-perfusion. The topography of retinal non-perfusion seems to be very important, both in the cross-section as well as deep, superficial and deep vascular plexus. Now, to do a trial in this area is really, really difficult because A, we don't know how to measure retinal non-perfusion. Secondly, we don't know what is clinically benefiting the patients with a change in retinal non-perfusion. Unless they affect the visual acuity, why do patients need to have all these injections? So these are all unanswered questions. Thank you very much.
It's an excellent understanding about the ischemia of in PDR. It's a pretty interesting studies what you brought it out. Any question? Yeah, please go ahead. Just use the mic. I have a question. I'll ask you later.
I am basically a glaucoma surgeon, so whenever we get NBG, the commonest procedure that we do, among the commonest ones, is valve which we put. So when we put a valve, the eye works like magic. All the NVE goes away, NVI goes away, and despite the patient being diabetic, he may live 10 years, 15 years. I have huge number of patients where we once put the valve, the NV just disappears. NVI doesn't come back. Very rare that I see it coming back.
Without PRP?
PRP is given. That is taken for
I
granted.
probably think the highest VEGF is in the aqueous. So I think when you manipulate your aqueous chamber, it does release all the VEGF. I think that might be the reason. I don't understand it. I don't know of any other reason. So hypoxic drive is definitely decreased. Is it?
I just a quick question about it, that when we did the study about the non-perfused areas, have you considered the pericyte loss into consideration while you're calculating it?
No, I will tell you OCTA by itself is extremely difficult. I mean,
Yeah, I understood, okay.
so to do a pericyte, I mean, we are still far off from that.
Okay, okay.
AO, like Amani Fauzi is doing quite a lot of work on AO. I think they are able to see the pericytes. I think it should be very, very specialized imaging.
Thank you, thanks a lot.
Thank you very much, Professor Silva Prasad. So wonderful. We have a great session, a lot of discussion. That's why we're running a bit late, right? Sorry about that. So we had to release the room. But then for our audiences and colleagues still interested in emerging research in ophthalmology, we can still have more discussion outside. So release the hall. Thank you very much. Thank you.
I think we should take a photograph outside. Please, we take a photograph outside, please.